Are you able to see the screen?
Yes, ma'am.
All right, Jaslyn, switch on your video. Am I audible to you?
Yes, ma'am.
Okay, on your video. And Aisha, you also see that if you are able to switch on your video or not, like try to rejoin the meeting.
No, ma'am, it's problem with my laptop cam. I'll
Okay.
rectify it and I'll see tomorrow.
All right, students. So tell me, have I done cryopreservation with both of you? Aisha, with you also,
No,
have I done?
no,
No?
no.
So what is the last thing that I taught to you? Aisha? Microsporogenesis was completed, right?
Yeah, that was completed. But we
Or
didn't write the notes.
you did not write. Only the
No.
microsporogenesis part, right?
Yeah.
All right. So after microsporogenesis gets completed, ultimately at the end, what is formed is the microspore or the pollen grain, right? Which is the male gamete. Now, in terms of their shape, they are spherical and they have a diameter of 25 to 50 micrometer. Now, every single pollen grain has been surrounded with two types of layers, the outer layer and the inner layer. Both of them have their characteristic feature. Both of them have their, you can say, stuff that they are made up of, and it's very different. So when we talk about the outer layer of the pollen grain, it is called as exine, and it is made up of sporopollenin. So the outer layer of the pollen grain, it is, you can say it is harder. It's very tough covering. Why? Because of the presence of sporopollenin. It's because the sporopollenin is very, very resistant, meaning that there is nothing that can degrade it. No acid, no alkali, no temperature, nothing can degrade sporopollenin. The exine of the pollen grain. Now see, for example, if this is the structure of the pollen grain, there is a part where the sporopollenin will not be present, and that part will be called as the germ pore. Okay, it is sort of like the opening present in the outer layer of the pollen. Now, because of the presence of the sporopollenin, the pollen grains are very well preserved. Because imagine having such a hard outer covering or the exterior. If it gets pressed under any rock or soil, it will not get degraded. It will not get destroyed. That is why, because of the presence of sporopollenin, the pollen grains are very, very well preserved in the form of pollen. Whereas when we talk about the inner wall, it is called as intine, right? It is thinner as compared to the exine, and it is continuous, means there are no interruptions in the intine. And the intine, it is made up of cellulose and pectin. Exine made up of sporopollenin, intine made up of cellulose and pectin. Now the cytoplasm of the pollen grain, it is surrounded by the plasma membrane because it is a cell. Every cell has plasma membrane present. Now, when you talk about the mature pollen grain, it has two cells in it. Other than the layers, there are two cells inside it. One is called as the vegetative cell or the tube cell. This bigger one that you see is the vegetative cell or the tube cell. And the smaller one is called as the generative cell. When we talk about the vegetative cell, it is bigger in size and it has abundant of food reserve because the pollen has to survive. So it has to have some sort of nutrition. And it has a large, irregularly shaped nucleus. We are talking about the first one. Then the smaller size one is the generative cell, and it is floating in the cytoplasm of the vegetative cell. It is not entirely round. It is spindle shaped and it has a dense cytoplasm and a nucleus. Now, one of the most important line that you people will ever come across is that in 60% of the angiosperms, meaning in majority of the flowering plants, the pollen grains, they will be shed at this two-cell stage, meaning the pollen grain containing the vegetative cell and the generative cell. This is very important to remember. Then what about the rest 40%? So the remaining species, in them, the generative cell will start dividing by mitosis and they will form two more cells. So the situation will look like this, like one cell from the vegetative, two from the generative. And this is called as a three-cell stage. So in the remaining of the species, three-cell stage is when the pollen grains are shed. Is it clear, students?
Yes, ma'am.
Any doubts? Aisha, is it clear to you?
Yes, ma'am.
Be responsive, students, okay? Because your videos are off, so be responsive. All right. Now, the pollen grains of many species, of many plants, they can cause severe allergies in some people, allergies and bronchial afflictions. And in, like you can say, severe cases, that can also lead to chronic respiratory disorders, meaning those respiratory disorders that will last a long period of time, such as asthma and bronchitis. What are those such pollen grains? So the pollen grains from the carrot grass, which is called as Parthenium. This came like a contaminant along with the wheat that was imported. This causes pollen allergy. Amaranthus, Chenopodium, they cause pollen allergy. But other than that, if you think about it, pollen grains are excellent reserves of nutrition, proteins and fats and all of that. So many a times, the pollen grain is also used as food supplements to provide more and more nourishment. So it is used in the form of tablets, it is used in the form of syrups, and the people mostly that consume this are athletes so that they can have their athletic performance enhanced, and also the race horses. Clear? Then comes pollen viability. Now, all of you know that pollen grains, they are shed from the anther upon maturation. Now, once they are shed, they have to land on the right type of stigma. If they don't, and they have to land on the right type of stigma much before they lose their viability. Viability means their effectiveness, their ability to fertilize the female. So the time period for which they are viable, they are valid, that is called as pollen viability. And this also varies from species to species, and it also depends on the temperature, the humidity, and the surroundings that the pollen is present in. For example, in the cereal plants such as the rice and the wheat, they are only viable for 30 minutes and that's it. After 30 minutes, not viable. But in the members of families such as Rosaceae, Leguminosae, and Solanaceae, here they are viable for months. So once they are released, if they do not land on the stigma, they'll stay viable for months and months. But for rice and wheat, within half an hour, they are done. Clear? Now, the last topic, as far as the male counterparts of the flowers are concerned, is cryopreservation. This is the technique to preserve the pollen grains. It's a method of storage. How do we do it? We store the pollen grains in liquid nitrogen at minus 196 degrees Celsius. Like we store it for future uses, we store it for crop breeding, just like how seed banks are present. Same way, pollen banks are also present. And this method of preservation or storage of pollen grain is called as cryopreservation. Clear?
Yes, ma'am.
Also, Jaslyn, tell me one thing. What is the last thing you have written in the previous class?
Ma'am, I finished writing cryopreservation.
You finished writing. All right. I want you both to be on the same page. So, Jaslyn, you wait for a while. Okay. Till Aisha writes it here. Okay. So, Aisha, now you can start noting down. Then we'll move forward and talk about the megascope.
Okay. Yes, ma'am. Yes, ma'am. You can just go. Ma'am, can you scroll left? Ma'am, can you scroll? Ma'am, can you scroll? Ma'am, can you scroll? Ma'am, can you scroll? Done, ma'am. Done, ma'am. Ma'am, I'm done with it. Yes, ma'am. I'm there.
Okay, any doubts up until now?
No, ma'am.
All right. Now see, now we have discussed about the male gametophyte of the plant. Now we will be moving on to the female gametophyte of the plant. That is the gynoecium. Now you people know that this is the female reproductive whorl of the plant. Now, one single unit of gynoecium is called as a pistil, right? Now, mostly in a flower, there can be one pistil or there can be multiple pistil. So accordingly, if a flower has just one pistil, we call that flower as monocarpellary. And if the flower has more than one pistil present in it, we call it as multicarpellary. Okay. Now, when the pistils, see, the different, if the several pistils are present, now they can be fused or they can be free. Like all the ends can be free or they can be fused. So when they are fused, we call them as syncarpous. This is found in the pepperwood plant. And when the pistils are free, do you see all of these free ends? That is called as apocarpous. So in biology, particularly for plants, the word syn means fused. The word apo means either it means at the top or it means free. So apocarpous kind of pistil is found in Mycelia. Clear? Now every pistil, it has three parts attached to it. Okay, there is the stigma, then the style, and lastly the ovary. Just like how the stamen had two parts, the anther and the filament. Here, the pistil, it has three parts: stigma, style, and ovary. Now, when we talk about the stigma, see, this flat surface that you see on, this is the stigma. It is the landing platform. So whenever the pollen grains are released, they travel either through the air or through the water or with the help of certain insects, and they reach the stigma. They land on the stigma. Then this long neck-like portion that you see below the stigma is called as the style. So the style is the long, slender part beneath the stigma. And what does it do? It connects the stigma to the ovary. And at the end, you see this basal bulgy part. This part is called as the ovary. Ovary is the basal bulgy part. And inside the ovary, ovules are present. But before that, inside the ovary, there is the presence of ovarian cavity, which you can also call as locule. And inside this locule or the ovarian cavity is the placenta. And arising from the placenta is the ovule. Now, this ovule is actually the megasporangia, just like how the stamen, the inside the anther, there was microsporangia, if people remember. So, in the female, there is megasporangia, which is formed from the ovule. So, this is the sequence that you have to remember. Ovary, inside the ovary, there is ovarian cavity, also called as the locule. Then inside the ovarian cavity, there is the placenta, and arising from the placenta is the megasporangia. Now, always remember that the fruit that we eat, right? Mango or you can say apple and all of that. So, in the fruit, the part that you eat, the fruit part is the ovary. So once the ovary gets matured in the flower, then that becomes the fruit. And you know that seeds are present inside the fruit. So that seed is actually formed from the ovule. So you can say that ovule develops into the seed or gives rise to the seed, and the ovary develops or gives rise to the fruit. Now you know that there can be like if you think about mango, so mango has only one seed. So that tells us that there was only one ovule present. But then there are fruits such as papaya, such as watermelon. They have multiple seeds, meaning they have multiple ovules in the ovule. Clear? Now, moving on to
Yes.
the megasporangia, which is the ovule. Now the ovule is of prime importance because inside the ovule only the egg will be made. And this egg will be very important for fertilization so that the male gamete, the pollen can fuse with it and form the zygote. So the ovule is a small structure that is attached to the placenta by the means of a stalk. Stalk means a stick-like structure called as the funicle. See, this is a picture of a typical ovule. So this funicle, this part, attaches the whole entire ovule to the placenta. Then the region where the body of the ovule fuses with the funicle, we call it as the hilum. So ovule and funicle, this part where it is fusing, we call it as the hilum. It's like a junction. Now the ovule, it's not present just like that. It is surrounded by protective layers. And the layers that protect the ovule, we call them as integuments. So there can be outer integument and inner integument. But there is one place where the integument is absent, and that place is called as a micropyle. So micropyle is that small opening inside, like that goes inside the this thing, ovule, or that helps the pollen tube to go. Don't think about the pollen tube right now. I will teach you later on. But remember that the part where the integument is not present is the micropyle. Now, always remember, even though this position is like this, right? You feel like this, everything that is at the base is at the bottom, but no, micropyle is the tip. So always remember that micropylar position is the top position. And the chalazal position is the basal position. So this image is actually inverted to think about the top and the bottom. But always remember this one thing, micropyle end, the top side, chalazal end, the basal side. So opposite to the micropylar end is the chalazal end, and this chalazal end represents the basal part of the ovule. Now inside the integuments, there are a lot of nutritive tissue, which we call as the nucellus. See this part? This part. It is the nutritive tissue, meaning it is rich in nutrients so that the developing ovule can have abundance of it. So that is the cells of the nucellus, they have abundant reserve food material. Inside the nucleus, we have the embryo sac, which we can call as the female gametophyte. Now the ovule generally has a single embryo sac, which is formed from the megaspore. Okay, so this is the structure of the ovule, just like how we studied the structure of microsporangia, if you remember. Inside the anther, we took out one cell, talked about the layers. Same way we are doing for the female part of the flower. After this, now this part is all the structures that you need to know. After this, just like how we have studied microsporogenesis, we will be studying megasporogenesis, meaning the process in which the megaspores are formed from the megaspore mother cell. In that, we saw the process in which microspores were being formed from the microspore mother cell. Right? Now, the megaspore mother cell, it is differentiated by the ovule in the micropylar region. So this megaspore mother cell is present towards the top side, right? Towards the micropyle. And it is a large cell that contains a very dense cytoplasm and a prominent means a showy nucleus. And then this megaspore mother cell, it starts to undergo meiosis to form the 4 haploid megaspores. But remember that out of the 4 megaspores, 3 are non-functional, only 1 is functional. Okay, only 1 megaspore is functional. So if you see in this picture, this egg-like thing that is present in the middle, to form this megaspore mother cell divides by meiosis. And this structure, this embryo sac is formed from 1 megaspore because the other 3, they will degenerate. Clear? Any doubts?
No, ma'am.
See, I have a picture. See this. See, the megaspore mother cell is there The megaspore mother cell, it undergoes the meiosis division and forms 4 haploid megaspores. Out of these 4, 3 are degenerated cells. They don't work. And only 1 megaspore forms the embryo sac. So this kind of development wherein the embryo sac is formed from 1 megaspore, we call it as monosporic development. Mono means 1, sporic refers to the megaspore, and development means development. So this type of development of embryo sac taking place from 1 megaspore, that is called as monosporic development. Clear? Any doubts?
Yes, ma'am.
Okay. See, this is like a very lengthy topic. So if you have any doubts anywhere, feel free to ask. Wherever you have doubts, we'll discuss. But for now, you people note down things here. How are you finding the chapter? Is it easy, difficult, medium?
It's
Yeah.
okay.
One by one. Jason?
It's okay.
It's okay. Okay, was it
Not
even
too
very
hard.
difficult?
It's not easy either.
And Aisha, what about you?
Same. It's not too hard, well, it's not too easy too.
And then when we talk about whether it is interesting or boring. Since it is botany, I know your answers, but still tell me.
Yeah,
Boring.
since it's botany, it's
Boring for both of you. How bad? No, say, ma'am, it's very interesting. It's so fun. It's like watching a movie. Say like that.
I think because it's the first chapter, it's not that boring. But yes.
Even if it is boring, you people have to learn everything. Okay, so you better make it interesting. Make some games, do something to make it interesting to you. Okay, or maybe I will do something. Okay, so up until now, I hope you people have no doubts.
Yeah.
All right, no doubt. Done, students?
Yes, ma'am.
You don't have to draw this picture. Just, you should know the example. You don't have to draw. Once you're done, do let me know by unmuting yourself, okay? This part is done, yes or no?
Yes, ma'am.
Aisha, you're also done?
Can you repeat?
I was saying that once you
Yes, ma'am.
people are done noting down, please let me know by unmuting yourself, okay?
Yeah,
Okay, ma'am.
yeah. Ma'am.
Yeah, this diagram you people draw. This is important.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am. Done, ma'am.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Done.
Done, ma'am.